Hello and welcome to Unit 7, Day 6. Today we're going to be talking all about mutations. So, how do mutations arise in an organism? That is today's aim. And in order to address this, first, let's refresh our memories. What exactly is a mutation? A mutation is a change in DNA sequence. And we're going to get a little bit more specific today and talk about types of mutations. But a change in DNA sequence. And this word here, arise, that just basically means happen. So how does a change in DNA sequence happen? And before we talk about mutations, let's take a look at this do now question. And we're looking at here, let's examine the diagram first. It asks, describe the main function of this process. And we can see here we've got the double-stranded DNA, and it looks like at the bottom here we can see these arrows. It's unzipping, and it's unzipping, and here at the bottom we can see there is an arrow coming up from the bottom, matching base pairs, and there's another one coming in this direction because that's the way DNA replication, oops, I just told you what we're looking at. That's the way this process happens. So we're looking at the creation of two identical strands of DNA, right? So we start with one strand, double helix DNA molecule, and we're here in the process, but we'll wind up with two identical DNA strands. So what is the main function of this process? Well, this process is DNA replication. We're copying DNA, and this is the first step of mitosis. The main function of this process is to make more identical cells. A mitosis, baby, and this happens in our bodies all the time. So in order for one cell to create two identical daughter cells, in order for this to happen, first we have to have DNA replication, and then those two identical strands of DNA can make their way into two identical cells. What would be different if the process pictured, if the diagram we were looking at were transcription? Well, transcription creates one single-stranded mRNA molecule. And that is all that would be created. So only one single strand of mRNA would be created instead of two identical strands of DNA. Question number 3, transcribe the following DNA sequence. So we have ATA, CCA, CTT. So let's transcribe that, baby, which means we're turning it from DNA into mRNA. So we have the mRNA sequence. Right, we start with the DNA sequence, and now the mRNA sequence. Going crazy with the colors here. U, A, U, G, G, U, GAA. All right, question number 4. Now let's use the genetic code chart below to translate. So now we went through transcription and now we're going to go all the way through to translation, the RNA strand from number 3. So we're going to take UAU, GGU, and GAA and translate them into their amino acid sequences. So let's go ahead and read this chart together like we did in class. So we're going to start with the first mRNA codon. And we have here our first base. So we're going over here on the left-hand side. And we're looking for the first base U. So we're in this row. And then we have the second base A. So in the top, we're looking at the second base A. So we're going to be in this third column. And then the third base is U. So we have the third base over here on the right-hand side. So we already have identified our box. We're going to carry it over. And our first amino acid is TYR or tyrosine. So then we're just going to repeat that process. For a little extra practice, you know, I'm going to show you myself on camera, but if you think you've got it, go ahead and pause this video and then see if you're right. But we're going to go ahead and take the first base, G. So we're in this bottom row. Second base, G again. So we're over here in this column all the way to the right. And then our third base, U. So we're going to follow that right there. GLY. And then our last triplet, our last codon, we're going to take our first base, G, in this bottom row again, our second base, A, so we're in this third column, and our final base, A again, we got GLU. So our amino acid sequence is TYR, GLY, GLU. This is our amino acid sequence. And now we're going to continue to use this universal codon chart for this entire worksheet. So first we're going to do some notes on mutations. So part A, mutations. And this first word on this first blank is going to be mutation. A mutation is a change in the sequence of DNA that is due to an error in replication, right? DNA gets replicated all the time. There could be an error, just like anybody could make mistakes. Your DNA can too. Environmental factors, like, for example, smoking or UV radiation, right? Too much sunlight can cause a mutation to your skin, which can result in skin cancer. So make sure you wear sunscreen. Such as certain chemicals, like again, nicotine or radiation, or even intentionally biologically engineered, like we saw, CRISPR can create intentional genetic mutations. But there are several types of mutations, and these names, I think, are going to be a piece of cake for you guys. Ladies and gentlemen, are you ready? So when a base is lost or deleted, we're going to call that a deletion. And we're going to look at examples of all of these in today's assignment. But that would be, let's say we have the amino acid sequence ATG, GAC, and when it is replicated, it makes AG, GAC. That would be a deletion. We could also have an extra base inserted. You want to guess what that's called? It's an insertion. That, for example, we have ATG, GAC. All of a sudden it becomes AAT, GGAC. And you may have already noticed what winds up happening. This changes what codons are being read, right? So just by losing one base or inserting one base, we can completely change how the line of code is being read because it affects what our codons are. And those triplets, those three base groups are very important because that's how our body actually reads these strands of RNA and DNA. So deletion and insertion may cause what's called a frame shift. Meaning it's reading different codons. So it may only be one base that changes, but as a result, the reading frame changes, meaning the groups of codons, and the entire amino acid sequence is affected after that base. So the frame shifts, and this can impact the entire amino acid sequence. All right, the next one is when a single base is substituted for another. And it's a substitution. And sometimes a substitution does not change the amino acid, right? Because it's instead of one base being inserted or deleted, it just winds up being switched for another or substituted for another. And this might not be that big of a deal. If it does not change the amino acid that is coded for, it is called a silent mutation. Meaning it may not even be noticed. It may go unnoticed. Because, hopefully you remember already, right? Multiple codons code for the same amino acid. For the same amino acid, meaning that the same protein would be made. And when we went over that, we called that mutation insurance, and this is what we meant. That because several codons code for the same amino acid, that means that if there's just a substitution, if it's ACC instead of ACU, you're still going to get THR. So it's not actually going to affect the final protein that is made. Pretty cool stuff. So now we're going to do some practice. So we have here the original DNA sequence. And I'm going to draw lines for our codons. And now we're going to transcribe the original DNA sequence into mRNA. And we're going to do this one together, but then you'll see for the rest of these, we're looking at a mutated DNA sequence. And what you are going to do is compare and highlight changes. Now, what you can do is you can actually pause this video and go through, and I bet you could do this entire page on your own, and then I'm going to go through the answers. But if you want to stick with me to get a little refresher, we'll do this first box first, and then you can do those other ones on your own, and I'm still going to go through all the answers. So, instructions. We're going to complete the mRNA sequences below. Use the chart above to find the amino acid sequence. So we're going to use the universal codon chart. Then you're going to classify each mutation as a deletion, insertion, or substitution, and whether or not the mutation is silent. So if it's silent, will there likely be effects? No, right? The silent mutation wouldn't have effects, but in the cases of a deletion and an insertion, yeah, there's going to be effects. And we're going to state the effects of the change there will likely be from this mutation. All right, so let's go ahead and do the first one together, starting with transcription, the easy part. So we're looking at TAC. That's going to become AUG, which is good. That's our start codon. ACC is going to become UGG. TTG is going to become AAC. GCG is going to be GCGC. Uh-oh, that's a mouthful. Easy to make a mutation error, but we're not doing the mutated DNA strands yet. ACG is going to become UGC, and ACT is going to become UGA. And now I'm not going to go through reading the entire universal codon chart again, but you're going to go up here and you're going to take AUG. So you can write them here alongside just like I did before. We'll do just one more as practice, but then you're going to do it on your own and I'll give you the answers. AUG, first base. I'm going to go over here, find it, first base, second base U. So we're here in this box. We're going to find G along the side, and it is our start codon. So we know we're going to start with AUG, then UGG is going to be TRP, AAC is ASN, AAC, and you want to get practice using this chart. So make sure that you are actually going through and using this chart and then just checking your answers with me. Otherwise, you're going to have a bad time later because you're going to have to use this chart on the exam. ARG, CGC codes for ARG, UGC codes for CYS, and UGA is our stop codon. So whenever a stop occurs, no other codons will be read. So there could be a million codons after stop, but they will not be read after the stop. Stop means stop. Drawing a little stop sign. Stop means stop. Do not pass go, do not collect $200. And start is the start of our sequence, and stop means stop. So we can see here, this is the original sequence, and then we're going to look at these mutated sequences. So pause this video, And it might take you about 10 minutes. We're going to do all 3 of these, and then you're going to press play, and we're going to go through it together, and I'm going to give you the answers. Okay, did you pause the video? I really hope you paused the video. So let's go ahead and look at this mutated DNA sequence. So comparing it right off the bat, I see one change. I see TAC the same, but ACC becomes ATC. That's different. TTG, that's the same. GCG, that's the same. ACG, that's the same. And ACT, that's the same. So this isn't going to be that big of a deal, right? Well, let's see what we actually coded for. So let's transcribe our strand. See if you got the same thing that I did. TAC is AUG. Again, that's our start codon. That's where we're going to start. ATC is UAG. TTG, AAC. GCG, CGC. ACG, UGC. ACT, UGA. All right, so if you go and look at the amino acids, you will find that we started with the start. And then even though all of these were the same, it doesn't matter because our second codon is a stop codon. So start, stop, that means no protein is made. That is a problem. Will there likely be effects? Yeah. No protein was made. Now, what kind of a mutation was this? Remember, basically the entire line was unchanged. We just had one C be substituted for a T. So this was a substitution. But it wasn't a silent mutation. Yes, no protein was made. So that's going to be a problem. Now let's go ahead and look at our second mutated DNA sequence. We have here TAC. And uh-oh, I'm already seeing something funny with this strand of DNA. So let's go ahead and separate this out into our codons. And we can see this errant T here at the end. This is no codon. Not codon, not a codon. So this is not going to code for anything. No amino acid. This is a frame shift. So if you compare this DNA strand to the original DNA strand, you see it's basically exactly the same. TAC, ACC, TTG, it's exactly the same. GCG, ACG, ACT, it's exactly the same except for this insertion, except for this G that stuck its way in there. So even though this is basically the exact same DNA sequence except for this one inserted base, the frame shifted. So the way our body reads the codons completely changes, and every single one is affected by this one insertion. Bad time. So let's go ahead and translate this bad line of code. TAC is still our start codon. So far, that's okay. Then now this U that's in there is a C, U, G. CTT becomes G, A, A. GGC becomes C, C, G. GAC becomes C, U, G. GAC again becomes C, U, G. And this errant T will become an A, but again, it's not a codon, so there's not going to be any amino acid there. So what amino acids do we get? We still have our start codon, so we're still going to start. And we're going to code for LEU, GLU, PRO, LEU, LEU, and then nothing. So this is making a protein. It's making some kind of a protein. It's not as bad as this start-stop where no protein was made. However, will there likely be effects? Yes. A totally different protein is made. So there's still a protein, but what the hell is it? I don't know. Totally different protein will be made. And what kind of a mutation is this? Well, we already did a little detective work, but we can see here that this G, that doesn't belong there, and it is evidence of an insertion mutation, which caused a frame shift. So even though it was just one base that was wrong, one base was inserted, that insertion changed the entire way that we read the line of code, and as a result, affected every single amino acid in that sequence, resulting in a totally different protein being made. Wowza. All right, so now this last one, we can do it. We're gonna separate out into codons. And we can see here, this looks very similar to our original strand of DNA. The first two codons are exactly the same. The third codon is a little different. The fourth codon is the same. The fifth codon is the same. And the sixth codon is the same. So we can go ahead and highlight our one change. We see that TTG becomes a TTA. I don't know why that made a little arrow, but just one base is different. One codon is different. And let's see what the mRNA sequence created is. TAC, again, we've got that start codon, AUG. ACC becomes UGG, UG. TTA now becomes AAU. GCG becomes CGC. ACG becomes UGC. And ACT becomes UGA. So our start and stop codons are still correct. And again, you're going to the universal codon chart every time, or you already did that, and now you're just checking your answers. And we have here in the middle, TRP, ASN, ARG, and CYS. And would you look at that? even though there was that substitution, we get the same amino acids. AUG is AUG, UGG is UGG, or rather, excuse me, AUG is start. The second one, UGG, is TRP, TRP, ASN, ASN, ARG, ARG, CYS, CYS, stop, stop. So will there likely be effects? No. The same amino acid sequence is produced. So what kind of mutation is this? This is a single substitution. And this is a silent mutation because the same amino acid sequence is made. This line of code codes for the same amino acid sequence, so the resulting protein is the same. Which is a relief for whoever had this mutated DNA sequence. There's no problems. So, all right, go ahead and complete your exit ticket. Today's aim was how do mutations arise in an organism? And we looked at deletions, insertions, and substitutions and did a good amount of practice. I hope you enjoyed that. Have a super fantastic day, and I'll see you guys all tomorrow on Zoom. Bye, guys.